There are over 7 billion people on the planet today, and we can notice a lot of diversity in our outward appearance, that is, our phenotype. And indeed, for centuries now, populations have been categorized according to their phenotypic similarities and differences, with the simple underlying assumption that populations that look more similar to one another are more likely to be closely related, as a result of a recent separation or interbreeding, than populations that look more different from one another. But reliance upon the phenotype alone can be misleading for many reasons, and systems of racial categorization carry a tainted legacy of oppression and violence. One's phenotype is the outcome of their genotype, that is their DNA, and the environment. So by looking at the genotype, we can often get a much more direct indication of the biological affinity of different populations. And indeed, over the past 30 years, this type of research has revolutionized the field of archaeology, bringing forth an understanding of past migrations and interactions that could not have been achieved with any other method. We are all one species, with anatomically modern Homo sapiens, us, having evolved in Africa some 150 to 200,000 years ago. A group left Africa around 50,000 years ago and eventually populated the entire world. So the incredible range of diversity seen in modern humans has occurred over this quite short time span. How do we know this? Both from the fossil evidence as well as from the clues contained in our DNA. Our DNA, our deoxyribonucleic acid, is contained in 22 autosomal chromosomes and a single set of sex chromosomes, XX for females and XY for males. DNA is our mechanism of inheritance, and for the DNA contained in the nucleus of your cells, you receive half of it from your father and half of it from your mother, except the Y chromosome, which is only inherited from the father and represents the male line. There's also a small amount of DNA on the cell's mitochondria, called mitochondrial DNA, which is only inherited through the female line, so reflects a population's matrilineal inheritance history. One's DNA encodes all of the information needed to build a human being. Specifically, each chromosome, as well as the mitochondria, contains specific genes which produce specific proteins. So, over time, DNA collects errors called mutation. These are then inherited. So quite simply, geneticists can compare the DNA sequence of one person to that of another person to see how similar or different their sequences are. Individuals who are closely related will share more of the same specific mutations than individuals who are more distantly related. When collecting genetic data for an entire group or population, the same principle applies. Populations that are more closely related will look more similar at a genetic level. So in this way, the collection of mutations stored in DNA is a record of the genetic history of a population. By way of a quick example, we can think of a mutation as a spelling error in the DNA code. It's been suggested that on average, any two humans will have about one spelling difference every 1,000 letters. The spelling differences can be thought of as molecular barcodes. People who share the same spelling difference typically share a common ancestor. Although we have to keep in mind these spelling errors can in principle also occur at different times and places independently of each other. For a short and clear summary of the genetic evidence for the spread of modern humans out of Africa across the globe, the timing and route of different migrations, we suggest you watch the slideshow put together by the Howard Hughes Medical Institute at the link shown on the screen. We've not yet bothered with the distinction between modern DNA, which is quite easily collected and replicated from any living human, perhaps from a cheek swab, for example, and ancient DNA, abbreviated as aDNA, which is what paleogeneticists try and extract from the remains of past organisms. The first study to report the successful extraction and sequencing of DNA from a non-recent organism came in 1984. And in that case, the DNA was extracted from an extinct South African equid similar to a zebra that had died about 150 years prior. That study was followed by several more by Svante Pääbo and colleagues, showing that this could also be done in human mummies that dated as far back as several thousand years. But ancient DNA is a lot more challenging to work with, and hence has developed into its own active subfield of inquiry, employing rigorous, time-consuming, and sometimes quite expensive methods that are well beyond what is needed when working with modern DNA. And that is because with time, the DNA molecule breaks down into smaller and smaller fragments, in the process accumulating post-mortem errors which can look like mutations. And it's also very easily contaminated by modern DNA, both from humans as well as from bacteria and other organisms in the burial matrix. Because of this latter concern, ancient DNA laboratories must be ultra clean and follow stringent protocols to prevent contamination by modern DNA or from the products of amplified ancient DNA from previously analyzed samples. There is a limit to how long DNA can survive for, with factors such as time, temperature, and free water being especially important. Earlier claims that DNA could be extracted and replicated from fossils that were millions of years old, say even from dinosaurs, have since been shown to be false. But many studies have successfully typed DNA from fossils that are hundreds of thousands of years old, still showing the tremendous scope of this technique. Many technological advances have been made that achieve a much higher chance of successfully sequencing even minute fragments of DNA, and also in detecting post-mortem alterations. These technological advances are beyond the scope of this video, but ample resources exist should you want to learn more. So, now that we've explained how DNA can be used to reconstruct an individual's or a population's genetic history, and about the challenges of working with ancient DNA, it's time for an example of this research. To tell us about her ancient DNA research on Dutch populations, we welcome Eveline Altena from the Forensic Laboratory for DNA Research at the Leiden University Medical Center in the Netherlands. Eveline, thank you for joining us today. I'd like to begin the interview by asking you about what spurred your interest in ancient DNA research, and especially how it is that you've come to be really an expert, a leader in the field in regards to ancient DNA research that's being done in the Netherlands.
Well, thanks, Andrea, for having me. As an archaeologist, I specialized in physical anthropology early on. And for me, ancient DNA research on human skeletons is basically an extension of that type of research. And what really interests me also is the combination between archaeology and biology, one of my two main interest fields actually. And how I got into this field is by an excavation in Eindhoven that took place in 2005 and 2006. And on forehand, we planned to collect DNA samples of all the skeletons that we would excavate. And in the end, that ended up into 400 skeletons being sampled. The date of these skeletons ranged between 1200 AD and 1815 AD, so that's a long time span. And that in principle enables us to look at population continuity for this specific population. And I worked as an archaeologist on the excavation, but I also did a PhD project that was related to it afterwards, to make a genetic profile of the Eindhoven population actually. That's what got me into the field.
So no doubt we can learn a lot from ancient DNA research, and one of the main aims that you've undertaken, and others as well, is to reconstruct population history, population continuity. So for the samples and the sites that you've looked at in the Netherlands, can you tell us a bit about that, and tell us why this type of research is important?
Well, Eindhoven at the time piqued a lot of interest, so because of that we were able to set up a new project similar to Eindhoven, similar period. And so in the end we were able to analyze almost 800 skeletons from 5 main sites all over the Netherlands. And that enables us to look at genetic variation not only in time, but also in space. And there are several reasons why this is so interesting. First of all, we know based on DNA research on modern population in the Netherlands that we have specific geographic patterns. And of course, we would like to know these patterns that we see. Are they of a recent origin? Are they of historic origin, or maybe even prehistoric origin? Yep. Well, unfortunately, prehistoric populations are hardly available for this kind of research because most of the skeletons that we find are very poorly preserved. That's a big issue, of course, in ancient DNA. So we can't really do population genetics on that level on prehistoric skeletons. We can do it on medieval skeletons. Mostly these are well preserved, good enough for what we want to do. So if you can see what the genetic variation of your populations are over time, you get a better idea of the history of that population. So do we see a lot of migration or not? Do we see bottlenecks, for example? But we can also look at selection, so several medically related variants. Do we see them change in frequency over time or not? Well, if you do see a change in frequency, it can mean that there was selection on these variants. But it can also be due to strong migration waves, for example. Right. So if you want to do this, what we call paleoepidemiology, you need to understand your ancient populations. You need to know what you're talking about.
Okay, so then a bit of a methodological question. Can you tell us what specific parts of the DNA molecule you've been analyzing specifically?
Well, we've been looking at mitochondrial DNA. That's very indicative for the female line in the population. The Y chromosomal DNA is indicative for the male line of the population, and the autosomal for the entire population. So all these three different parts of the DNA, so to say, together form a very coherent story of the population. Of course, mitochondrial DNA is most abundant in our cells, so considering degradation that takes place on ancient DNA, these always have the highest chance. And overall, on these medieval populations, we have a success rate of around 90 to 95% for mitochondrial haplogroup typing, which is, of course, quite high. And that also really does enable us to look at variation in time and space, because you need the information to do that, of course. But luckily, our populations are well preserved.
Indeed. So then the final question has to do with the results. Now, I know a lot of the research that you've done is in the process of being published as we speak. So there's only so much you can share with us today. But if you could perhaps share with us some of the highlights of what you found, or something that you found particularly interesting or striking in terms of the results.
Well, due to relatively recent technological advances, we are now able to look at ancient skeletons on a population level, and that means that we can look at population continuity or maybe even discontinuity. But for that you need specific and sophisticated statistical tools, and these are being developed at the moment, and we can now apply them to our populations. That's very exciting, because before what you could do is just compare different populations from different periods, but you could never introduce the factor time in your statistical analysis. And of course, time is a very important factor. And so by introducing that factor in our statistical analysis, we can see if frequency changes of our variation occur due to natural processes, such as drift, for example, or more to significant migration waves, or maybe even bottlenecks. That's exciting for us to be able to apply these new statistical models. But what I also saw is that several Y chromosomal haplogroups that we see in very, very low frequencies in the modern Dutch population, so that we consider as exotic, actually, to our surprise, were already present very often already centuries ago, and that was really not something that we expected. So
interesting. Yeah. Well, speaking for myself, and I'm sure many of our viewers, I look forward to hearing and reading more. And thank you for taking the time to join us today.
You're welcome. It was a pleasure.
In this video, you've learned about the methods to reconstruct the migration history of a population using genetic data, that is, our DNA. Both modern and ancient DNA contain clues about our evolutionary history, allowing paleogeneticists to trace the migrations and movements of the world's populations, past and present. You learned about ancient DNA research in the Netherlands and what it has revealed about populations who lived, and in some cases are still living, in this area from the past 2,000 years. Coming up next is a short video where the teaching assistants and I will briefly discuss some of the big questions about human mobility and migration that were mentioned in earlier videos. We'll see you there.